A very good morning, all of you. In last lecture, I have taught about the structure of nephron. Nephron is nothing but it is the structural or the basic unit of the kidney where the filtration of blood takes place, and these filtrations of blood is very essential to separate, to filter the unwanted substances which is present in that blood. Later on, in today's lecture, I am going to talk on urine formation or the process of urine formation or the mechanism of urine formation. Now, the urine formation is the process or a step where the formation of urine takes place, and this urine formation is completed in the 3 steps. These 3 steps are the filtration, or it is also called as glomerular filtration. Second step, reabsorption, and third step is called as the tubular secretions. The kidney filter unwanted substances from the blood and produce urine to excrete them, and these urine contain the nitrogenous waste products. Now, especially the formations of urine is a very complex process. It is completed in these 3 steps, which is called as, in this microphotograph, it is very clear that how the urine formation takes place. We are knowing that the first steps, especially the first part of the nephron, which is called as it is nothing but the Bowman's capsule, and it is a glomerular. Now, these Bowman's capsule or the glomerulus act as it is a fine network of the blood vessels which is entered in that glomerulus, called as afferent arterioles. These afferent arterioles possess the bigger size, whereas from that afferent arterioles there is the number of network fused together to form a very mesh-like, net-like structure of that blood vessels, and these act as filtrations of the bloods. These vessels capsule, which is called as a glomerular capsule, or it is a specific capsule which is essential for the purpose of the filtrations of the blood. Now, the next process is called as a tubular reabsorptions. Especially these tubular reabsorptions, takes place in the PCT, proximal convoluted tubule. PCT is nothing but the proximal convoluted tubule, where the reabsorptions of some important material which is excreted in the glomerules. Such material reabsorbed by the specific tubule, which is called as a tubular reabsorptions, and it takes place by the PCT, proximal convoluted tubules. Then the secretion, which is also called as a tubular secretion. This tubular secretion is also the part of the urine formations, where it generally balancing the ions as well as the water in that specially excreted material, and lastly it opens with the collecting duct via the urine created. Now the first step, which is called as ultrafiltration, the formations of the kidney filtrate, where the filtration mechanism is takes place. We are knowing that the Malpighian body or the glomerulus is the locations of ultrafiltration. The glomerulus increases blood pressure. How it is increases? It increases blood pressure by forming a narrow branches of the blood vessels, which is also an increase in surface area for the filtration mechanism. Means it also increases the surface area for the process of filtration. The pressure is maintained by the narrower different arterioles, especially which restricts the outflow of blood. Means it is a very special phenomena, a special mechanism there takes place. The entry of blood with the wide tube, which is called as the afferent arterioles, but the exit of blood takes place in a narrow blood vessel, which is called as the efferent arterioles, and the expanded end of the nephron form an invaginations to form a cup-like structure in that glomerulus, and the efferent blood vessel associated itself with the other region of the nephron for the selective reabsorption. Now, this is a very clear structure about the glomerulus, how the process of filtration there takes place, which is called as a glomerular filtration. Now, this is the afferent arterioles. The arterioles having the wide size, it is afferent arterioles. These afferent arterioles becomes a very thin, meshy or mesh-like structure of the vessels capsule, which is called as a glomerular capsule, and these capsule rejoin to form a specific type of arteriole, which is called as a efferent arteriole. Means at one end it joins with the afferent arterioles, and other end it joins with the efferent arterioles. And this is the sites of filtrations, and via these generally the filtered materials they are collected in this, especially PCT, proximal convoluted tubules. Now the formations of urine or in ultrafiltration, especially this stage, it is very clear that what filter via this glomerulus. There is a filtrations of water, there is a filtrations of mineral salt, filtrations of glucose, amino acid, as well as the nitrogenous waste product. And out of them, the mineral salts, water, glucose, amino acid is a very important, and it is reabsorbed by the further tube-like structure of the nephron. Especially the ultrafiltration takes place through the three layers. Means the glomerulus is having the specific type of layers, a cup-like structure. These three layers is endothelium of the blood capillaries. This is the endothelium of the blood capillaries. The basement membrane of the blood capillaries. These are the basement membrane of that blood capillaries. And here, this is the sites of the epithelium of the Boehm's capsule. And generally, via these three layers, the ultrafiltration is generally takes place. Then the second step, reabsorptions. Reabsorptions, it is the movement of substance phase out of the renal tubule back into the blood capillaries. Means those efferent arterioles, they continue along with that renal tubules, and these blood vessels generally by the process of diffusion, it is reabsorbed by these important material which is filtered in that renal tubules, which is called as the reabsorption. The substances reabsorbed are which substances? Water, glucose, other nutrients, and sodium as well as the other ions. It is also called as a selective reabsorption because it is selectively reabsorbed. These important material which is passed in that tubule. The process of control and regulation in the kidney begins with the non-spatially discriminating filtration, which is called as the ultrafiltration, that remove just as many useful substances. Means along with the harmful substances, some useful substances may also filter, and these such important useful substances is absorbed, which is called as the selective reabsorption. The kidney then takes back from the filtrate to the blood those substances that it still requires in the blood. The beauty of the way of kidney works is that it is able to control how much substances it reabsorbs back into the blood. It is called as a regulation. Means it regulates which substances is essential. Such substances may be reabsorbed. Means especially the process of reabsorptions, they are takes place in this way. Here the only water is reabsorbed, but ions reabsorb here. Means the ascending and descending loop of Henle, these LH loop of Henle, descending and ascending, it absorbs some places water, it absorbs in some parts it is important ions, and generally the PCT means especially these parts, it also permeability to water as well as the salt is regulated by the hormones. Means the hormones is also very essential to involving in the process of that reabsorptions in the nephron. The selective reabsorptions, especially the process of reabsorption, they are takes place in that PCT, proximal convoluted tubules, all sugar, most salts, and some water they are absorbed in this part. In the loop of Henle, the water potential of the fluid is decreased by the addition of salt because the salt they are entered here, and due to that this is the decreased additions of salt and removal of water. But here the water potential increased as salt are removed by the active transport. This is the specific mechanism via this it is a regulated water salt balance. Generally the water potential decrease again by the removal of water, ensuring that urine has a low water potential. Means especially it is completely balanced. In urine there is also water content is essential, and all are regulated via these three process, as well as it is also called as a pin mechanism, which is also called as a counter current mechanism. The most of reabsorptions occur from the proximal convoluted tubule, where the 85% of the filtrate is absorbed. Means most of the absorptions they are takes place in the PCT, proximal convoluted tubules, and all glucose, amino acids, some salts, and water are reabsorbed in this process. The reabsorptions of the water as well as the salt, they are take place in this PCT. 65% of the salt and water. Means in some books it is noted that the number percentage is varied. That is the from 65 to 85, and the another remaining reabsorptions they are take place in the LH loop of Henle. Especially this sketch is very clear that here the filtration mechanism is takes place. After the filtrations, this part is essential for the selective reabsorptions as well as the secretions. And after that, via the renal vein, the excretions is there takes place. Means completely excretion is nothing but the filtration, reabsorption plus secretion. Completely it is called as a process of excretion. The third is the tubular secretion. Tubular secretion is the transfer of material from peritubular capillaries to the renal tubular luens. It is an opposite process of the reabsorption, and this process or the secretion is caused mainly by the active transport and passive diffusion. For the process of diffusion, for the process of especially the transports, this secretion process is very essential, and usually only a few substances that are secreted in this process. The active tubular secretion, it is also called as a drug, are pumped from the blood to the tubular urine via this active transport system. Means it is an area of the lower concentrated towards an area of higher concentration. Means the transportations, they are also take place in the same direction. It is from the lower to the higher concentration level, and it for that purpose it requires the energy. The tubular cell, which contain the specific type of the glycoprotein, P glycoprotein, in their membrane, which is act as a pump drug into the urine. This is the specific mechanism. In the tubular secretion, it is the process in which the renal tubule excretes or extracts the chemicals from the capillary blood, and it secretes them into the tubular fluids. If those unwanted material, those extra material, they are becomes present in that blood, it is secreted in that tubular fluid, in the PCT, proximal convoluted tubule, and the nephron, as well as the loop of Henle, the tubular secretions, they are takes place. What is the purpose of these tubular secretions? It is essential to remove the balanced waste material. It also regulates the acid-base balance. Hence, it is very important. Especially the secretion, it is generally takes place in the proximal convoluted tubule as well as the distal convoluted tubule. Secretion is active as well as the passive. The passive secretion depends upon the pH. And what is secreted? Which substances to be secreted? That is the ions, the potassium, ammonia, hydrogen, as well as the drugs and some xenobiotics. These substances becomes removed, and generally it is created via this process. After the process of the urine formation, the last bit of this chapter is the counter current mechanism system. The counter current mechanism system is a mechanism that expends energy to create the concentration gradient. And for that example, means for the example, it can refer to the process that is underlying the process of urine concentration. That is the productions of the hyperosmotic urine by the mammalian kidney. Especially this counter current mechanism is also called as, it is a hairpin counter system or hairpin counter current multiple theory. This theory was proposed by the scientist Wirtz in 1951 and Bray in 1960. They explain the formation of the concentrated urine. This system operates in the hairpin-like loop of Henle. Especially via the hairpin-like loop of Henle, this system they becomes operated. It is a system in which the flow means inflow runs parallel to counter to the, or especially it is a system in which the inflow runs parallel to counter to and in close proximity to the outflow of the some distance. The conditions to be fulfilled for that counter current mechanism is two tubes in the parallel, movement in opposite direction, in close proximity, and selective permeability. The counter current mechanism in kidney, it is depend upon maintenance of the gradient of increasing osmosity along the medullary pyramids. The counter current multiplication system, they are generally takes place in the loop of Henle. The counter current exchange system, they are takes place in the vasa recta. The long loop of Henle generally establish a vertical osmotic gradient where the counter current multiplier they are takes place. Their vasa recta preserve or preserve this gradient while providing blood to the renal medulla. Means especially the exchange of counter currents they are takes place. It is called as the exchanger. The collecting duct of the all nephron use the gradient in conjunction with the hormone, especially here the hormonal role is also essential. The vasopressins or the ADH antidiuretic hormone to produce urine of varying concentrations depending upon the specially the osmotic equilibrium. Collectively, this entire functional organization is also called as a medullary counter current system. Means in this case, in the loop of Henle, how the counter current mechanism they are takes place, how the especially the active transport as well as the passive transport they are takes place. The active transport they are takes place with the red arrow, while the passive transport they are takes place with the blue arrow. This is about the means the especially the passive transport of the water they are takes place via these blue eyes, whereas the some ion salts that is the NaCl here in this place it is also the active transport, but later on especially in the vasa recta these NaCl and H2O they are generally move opposite directions via the passive transport. This especially this is nothing but the in the descending as well as the ascending limbs, the renal fluid flow in the opposite direction and hence the name counter current or current counter. Means it is generally move in opposite direction, and this mechanism of this theory to produce concentrated urine can be proceed, can be formed, and especially the entire length of the nephron is the permeable to water. Means overall the tubule of the nephron they are accepted to specially water permeable, and with the exceptions of the some ascending limb of the loop of Henle, sodium is the actively pumped from the ascending limb to the intestinal fluid, that is the medulla. Especially later on, this is the key fact that the descending loop of Henle, LOH is nothing but the loop of Henle, is a water permeable. The ascending loop of Henle is not a water permeable. The ascending loop of Henle is act as an actively pump out the ions. Hence, the water goes to where the most stuff is, and the vasa recta remove water so it does not dilute the medullary gradient. Especially, it is generally regulated. These are some important feature of the concentrating process or the counter current theory. Especially the descending limb of the loop of Henle has a high permeability to the water and low permeability to the solutes, salts. The ascending loop of Henle has a low permeability to water but actively reabsorb the sodium ions. The distal tubule and collecting duct also actively reabsorb the sodium ions. In the presence of ADH, antidiuretic hormones, they are permeable to water. And the descending and the thin ascending limb of the loop of Henle are the permeable to the urea. The thick ascending limb and cortical part of the collecting duct have a low permeability to the urea. Means here, this is the explanation about the part of the nephron where permeability of water, permeability of the ions, permeability of the urea, they are takes place. The medullary part of the collecting duct has a high permeability to urea that can be increased by the ADH, antidiuretic hormones, especially. Now, overall, especially the so many salient feature of this theory is the ascending limb actively transport the sodium ions. The medullary tissue is concentrated. In this way, they balance the water ion proportion concentration in the urine. Hence, it is called as a countercurrent mechanism. In this way, today's lecture, I have summarized about how the urine is formed, as well as what is about the countercurrent mechanism. When the urine is formed, basically the urine is yellowish color. Because why urine is yellowish color? In so many examination or in viva voce, it is asking that the urine is yellow in color due to it is having the urochrome, especially, and especially in about the 1 to 1.5, 1.8 up to the 2 liter urine, they are excreted by the normal persons, and these urine contain the most of the some organic as well as the inorganic substances. That is, it means the urine is having the salty stress. It is a salt-like structure. It is acidic in nature. It is having the hypertonic, aromatic odor, and generally this urine is contain the urea, ammonia, uric acid, creatine, hypuric acid. These are the some content compositions of the urine, as well as besides that some amount of vitamin C, oxalic acid, as well as the glucose and albumin is also contained in that urine. Overall, it is about the countercurrent system process of urine formation as well as the compositions of the urine. Thank you very much. Is it any doubt about the overall chapter? We are having the 4 to 5 minutes. Vanchewad. Vanchewad. Assignment. I am also giving the assignment in 1 or 2 days. No problem. Vanchewad. Unmute yourself. क्लास चालू आहे. क्लास चालू असताना